right, thank you very much. So my name is Carlos Garcia, and I am a Smithsonian Institution postdoctoral fellow. And well, this is a project that I am developing in collaboration with the Departments of Botany, the Department of Entomology, and also with the Encyclopedia of Life. And well, we all know that human activities are generating species extinctions, and the main processes generating species extinctions are habitat loss, species invasions, overhunting, and, well, here involved climate change. And it is estimated that by 2050, around 15% to 35% of all the known species on Earth will be at risk of extinction. Well, however, this situation can be even worse. Here are some examples of species that are depending on other organisms. So we have, for example, plants and their insect herbivores. We have fig trees and their pollinators, the fig wasps. We have birds, mites, and all birds, primates, and all their associated mites, lice, and nematodes. And if these species disappear, then all the associated species to them will also disappear. And it is expected that if this happens, maybe the order of magnitude of extinctions will be one order of magnitude higher than what we know so far. So here we have some examples of these interactions. And what we have here is the expected proportion of coextinctions after the other organisms disappear. And what we see here is that when you have closely related organisms, they will disappear. But if the interactions are not that specific, then you will have a lower rate of extinctions. Well, one issue is that the current extinctions estimates don't include coextinctions. Therefore, current extinction estimates need to be recalibrated by taking species coextinctions into account. So today, I will talk about two challenges and solutions estimating risk of coextinctions using as example plant-herbivore interactions. So first, how to identify the species interacting, and second, how to actually be able to identify the interactions between those species. So we're performing this research at the Selva Biological Station in Costa Rica in Central America. And we're performing this study in the Barva National Park, that is the highest elevational gradient of continuous forest in Central America that goes from tropical rainforest to mountain forest. And we are using a study system, Cephalolea beetles, and plants from the order Zingiberellus. And these are really particular interactions. This is one of the oldest and most conservative plant-herbivore interactions. These beetles and these plants evolved together for the last 60 million years. And what we have here is a map of the study sites along the elevational gradient. And what we're doing here is we're going to which one of these elevations, and in each one of these locations, we are recording the altitudinal distribution of each insect herbivore species and each host plant. So to identify insect diets, we are using DNA barcodes. So first, what we do is to obtain DNA sample for each one of all the potential host plants of the beetle species and generate a host plant DNA barcode library. Then we extract DNA from the insect herbivore, and then by comparing these two sequences, we can actually match the diets of each one of these different herbivores. All right, and well, all the morphological data and all the natural history data is included in the Encyclopedia of Life, and all the molecular data that we're generating goes to the gene bank. And also, EOL and the gene bank are connected. But what is really important of this project is that all this information is used by taxonomists together with all our collections to be able to identify each one of these species. So taxonomy has to be really stable if we want to understand coextinctions. Here are some results. So these are the networks of interactions of plants to your left and beetles to your right at different elevations. And so far, we have all the interactions of plants and insect herbivores at two elevations in this mountain, tropical rainforest, pre-mountain forest, and right now we are doing the same kind of sampling for the two higher elevations. So now a little bit about the big picture. So insect herbivores with more specialized diets are more prone to coextinctions. And, well, these are bad news because most insect herbivores are specialists. 95% of all species of insect herbivores on Earth are specialized. Second, insect herbivores are more prone to coextinctions at higher elevation. And, well, this is also bad news because climate change will first affect high elevation ecosystems. Okay, so the next step of this project is to use a digital elevational model of the Barva transect, that is this mountain in Costa Rica that is to your left. And what we're doing right now is we're modeling the extinctions of host plants. So we can model the biological attritions, the range shift, but also the extinctions of the host plants. And then with this base information, we can model the coextinctions of all the associated plant herbivores. Thank you very much. Right, so the question is, how are we modeling the cascades of extinctions in this mountain? Well, there are two ways of modeling this. One is the approach that we're using for these data sets, where we are actually removing in simulations each one of those links that I showed in this presentation, and then with this you can estimate a risk of extinction depending on the diet breadth. That's one way of doing it. Another way of doing it, and that we are doing it too, is with alternative experiments, where we are measuring the extinctions using the population growth. So you can actually, this is a different way of measuring extinctions. You have a modeling of extinctions at the community level using links, but you can also measure the NDT, the changes of population growth in populations at different temperatures.